so let's talk about some biological molecules and compounds that we find in the body. So, we are made up of two kinds of compounds. The human body has either inorganic or organic compounds within it. Organic meaning carbon-based. So, one of the major inorganic compounds is water, right? Water is H2O. It does not have carbon in it, hence why it is inorganic. It composes 2/3 of our body weight, and it absorbs and releases large amounts of heat without much of a change in temperature. Water has some really amazing properties, and that is what allows life on Earth to continue, basically because of water. It's amazing. And then we have, water is one of the main solvents for many solutes. Solutes are the things that dissolve in the solvent. So if water is the solvent, maybe salt is the solute, or sugar is the solute. And water is the site of many chemical reactions within our body. It serves as a lubricant. It assists in the movement of food through our digestive system. And it is found in many different places. It is in the extra and intracellular fluid. It is in our tissues, it's within our plasma, in our lymph, and in other specialized fluids. We need to talk about what the difference is between an acid and a base and the pH scale. Acids increase the hydrogen ion concentration of a solution by releasing H positives, hydrogen atoms. Bases decrease the concentration of H by combining with the H already in the solution. And then we have this thing called the pH scale. And the pH scale is the measure of the hydrogen ion concentration in any given solution. So, the pH scale basically indicates the acidity or the alkalinity of a solution, meaning the concentrations of H plus or OH negative. H plus denotes an acid, OH negative denotes a base. The pH scale runs from 0 to 14. 7 is neutral. Anything less than 7 falls to the acidic side. Anything greater than 7 falls to the basic side. Our bodies, for the most part, hover around 7, neutral. There are some things in our body, like stomach acid, that is much more acidic than 7, but our tears, our saliva, our urine, it's all close to 7. Here is a pH scale with some examples of different substances and where they fall on this scale. So hydrochloric acid is a 0. That'll burn your skin off. There's your stomach acid, some lemon juice, vinegar, tomatoes, black coffee, urine, saliva, kind of, 6-ish depending. Then we have 7, which is neutral. Then we go the other way. Seawater, baking soda, ammonia, oven cleaner, and we end with sodium hydroxide. Okay? So it just depends on the concentration of OH negative or H positive. And that concentration will make the substance either acidic or basic. There are things called buffers, and they are chemicals that can either pick up or release an H+, and therefore they can keep a solution's pH constant. because they're buffering the system. That makes sense, right? We have lots of buffers in our body, and this is what allows our pH to stay relatively normal within our body fluids. And we need this because even slight variations of pH can be very harmful to our cells. So we have lots of really great buffer systems in our body so that we can maintain our homeostasis, right? Then we have some organic compounds. One of them being carbohydrates. Carbohydrates are formed of carbon, oxygen, and hydrogen. And you find 2 H's for every O. 2 hydrogens for every oxygen. That's just how they're set up. That is part of being a carbohydrate. They are a primary source of nutrient energy for our body cells, and they can be classified depending on how they're set up. So we have monosaccharides, disaccharide, and polysaccharide. Saccharide meaning sugar, mono meaning simple, monosaccharide, simple sugar. They are the simplest of carbohydrates, such as glucose, fructose, and galactose. Okay? We talked about there's 2 hydrogens to each oxygen. Here you can see that in glucose. You have 6 oxygens and 12 hydrogens. That is a 2-to-1 ratio. You always find that in a carbohydrate. There are disaccharides, such as maltose, sucrose. Sucrose is table sugar, meaning the white sugar that you might put in your coffee or in other items. That is sucrose. And there is lactose. Then we have polysaccharides that are basically formed by chemically combining lots of chains of monosaccharides together. One of them is glycogen, which is how animals store carbohydrates. We store glycogen. And you also have starch, which is how plants store carbohydrates. Another category is lipids. Lipids consist of carbon, hydrogen, and oxygen. There is usually a backbone of carbon with many more H's than oxygens. They are abundant in the body, and we find triglycerides, phospholipids, which we find in the cell membrane. We will talk about that when we get to the cells. And steroids are actually a type of lipids or fat. Triglycerides are the most concentrated energy source in the body. They are around internal organs and under the skin. They are hydrophobic. Hydrophobic meaning water hating. Hydrophilic, water loving. So fats don't like to be around water, right? They don't dissolve. I'm sure you've been cooking or cleaning at one point, and the fat maybe, or the grease on the pan doesn't dissolve in the water, right? It repels, and that is because they are hydrophobic. 1 glycerol molecule and 3 fatty acid molecules join together to make what we call a triglyceride. And there is a picture of it for you. And it's those fatty acid portions, those tails, that are so hydrophobic. Then we have things that are called saturated fats or unsaturated fats. Sometimes people say the saturated fats are the bad ones, like the butter or the lard. Those are things that we find solid at room temperature, whereas an unsaturated fat is usually a liquid at room temperature, and it would be more plant-based, such as olive oil or corn oil. And that has to do with how the H's are stuck onto those carbons. If there's no more room for H's, it is saturated. But if there is a double bond between some of those carbons, then it is unsaturated, meaning that those double bonds could be broken and more hydrogens could be stuck in. So I'll tell you a little story. If you've ever had peanut butter, okay, if you've ever bought a jar of peanut butter from the store, and you bought like Skippy or Jif or something, it's real creamy, the peanut butter, right? It's really creamy. But if you've ever had natural peanut butter, it kind of separates, right? You get like a layer of oil almost on top of the peanuts. And that is because the peanut butter that is processed or not natural isn't as good for us as the natural peanut butter, because it is what we say, it is hydrogenated, meaning those double bonds have been broke and more hydrogen has been stuck in to give it that creamy texture that some of us really enjoy. But by doing so and chemically changing it, it isn't as good for us, right? So if you really love peanut butter and you want to have the best peanut butter for your body, you're going to have to stir that oil back in. All right, then we have phospholipids. These, like I said, are found in the cell membrane. They are composed of one glycerol, two fatty acid, and one phosphate group. They are hydrophilic, meaning water-loving. And it depends on the tail and the head and how they associate to water, and we'll talk about that when we get to the cell membrane. They all come together in a certain way because they are hydrophilic and actually hydrophobic at the same time. The head is actually hydrophilic, the tail is hydrophobic. Here's steroids. They are molecules consisting of 4 carbon rings. Cholesterol is a type of steroid and a cell component, and we find it in the cell membrane. There are chains of cholesterol that allow for flexibility of the cell membrane. And then we have another category, and that is a protein. Proteins are made up of amino acids. There are 20 different amino acids in our body, and they each differ in their R group. So basically, they all have carbon, hydrogen, oxygen, and nitrogen. That's the same. But then they have this specialty group, and that makes them special. That gives them their individual properties. So it can either be an amine, an acid, or an R group. So at the bottom here, we have some pictures of just, the first picture is just a general amino acid. That's going to show you some carbon, hydrogen, oxygen, and nitrogens, and a special R group. And then as you move down the line here, you can see the different types of amino acids depending on what's in that green box. Depending on the difference of the R group gives that amino acid its individual properties, its individual reactions and characteristics, and those all play a big role in our body. You've all heard the terms like, the cereal contains 7 essential amino acids or whatever. We need amino acids. We don't make them all, so we need to take some of them in a healthy diet. And they differ literally depending on that R group, which is what you see in the green. They are joined together by peptide bonds, either dipeptide meaning 2 or polypeptide meaning many. Each polypeptide or protein has a unique 3-dimensional shape. And proteins are classified either structurally or functionally. And proteins do take on specific shapes according to what they contain. And that is that unique 3-dimensional shape. And if they lose their three-dimensional shape, we say that they become denatured. And if a protein becomes denatured, it does not work anymore. And that's a problem. We do not want the proteins in our body to become denatured. That is an issue. Enzymes are a type of protein. And enzymes basically catalyze chemical reactions. They speed up reactions. They help our body do things as effectively as possible. And there are substrates and products, and there's sometimes what they call a lock and key fit. So enzymes work with only certain substrates, and it allows our body to perform functions at a very quick rate and a very effective rate. But again, because an enzyme is a protein, if the enzyme loses that 3D shape, that specific shape, it cannot function. Again, problem, we want our enzymes to be functioning as well as they can, because they help in many chemical reactions in our body. And lastly, the last group are nucleic acids. There are two types of nucleic acids. There is either DNA or RNA. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. They are the building blocks of nucleotides. Basically, nucleotides consist of a base, a sugar, and a phosphate. And this is where we get those names: adenine, thymine, uracil, right? In DNA, you have A and T, C and G, and in RNA, you have AU, we swap out thymine for uracil, and CG. And that's what makes up, who we are, our DNA, right? It's all composed, I'm sure you've seen the letters and how they match up, A with T, G with C. Here's a picture showing us the sugar phosphate backbone, and then we have those nucleotides coming across the ladder, if you want to call it a ladder. And they always pair the same. If they don't, there's a problem. That could cause a mutation. So you always have the sugar phosphate backbone, and then you have the nucleotides in the middle, the two strands. And those are hydrogen bonds between them. A and T have 2 hydrogen bonds, and G and C have 3 hydrogen bonds. And that's the dashed line that you see there between the letters. And it makes kind of a ladder shape. Sometimes it's called a helix. RNA is a single strand, and it, as I said, the thymine is swapped out for uracil, and it has different functions. RNA deals with protein building and things like that. So DNA is double-stranded, RNA is single-stranded. Then we have something called ATP, adenosine triphosphate. Tri meaning three phosphates. So it is a modified nucleotide with an adenosine and 3 phosphates. And basically what happens is it has the ability to store energy between the phosphate bonds. So when you pull off, when your body basically synthesizes one of the phosphates off, it can use the energy that was in that bond. So ATP can be broken down into ADP, which is diphosphate, meaning 2, because you've taken off 1 phosphate, and that gives us energy. There's a picture of it. So we have the A the adenosine there with the 3 phosphates, and then you pull one off and you get a burst of energy, leaving you then with ADP and an extra phosphate with energy. All right, that is going to conclude our lecture on inorganic and organic molecules and compounds. Please let me know if you have any questions.